All right, so as Gurleen said, hello everyone, welcome back from lunch. I hope you had a nice break and are now really ready for the camp's first workshop, which is presented by Simply Neuroscience's Brain Resource Creation. So let's get right into it. So, Brain Resource Creation, or BRC, is a department of Simply Neuroscience that creates educational resources for the Brain Bee competition, which, if you guys don't know, is a neuroscience competition for high school students. And today, BRC is here to teach you about synaptic plasticity and the rate of learning.
Yep, so this workshop here has been created by Erin, and Erin's also here with Pavan to present it. So you guys are actually getting to learn the information right from the creator of this awesome workshop. We're going to ask that you keep your audio and video off during the presentation, just because it is going to be recorded, and we want to show our respect to our presenters. But now, without further ado, let's actually get started.
All right, hello everybody. My name is Erin, and I'll be presenting this workshop along with Pavan. I'm coming at you from Los Angeles, so pretty nice outside today, very sunny, and very excited to be here. And I'll pass it over to Pavan.
Hello, I'm Pavan. I'm from Massachusetts, and yeah, the weather is not great here, but it's still a good state.
True. Okay, so jumping right into the content. So this workshop in general covers, and I apologize for a bit of background noise as well if you ever hear anything like that, but so this workshop will cover synaptic plasticity, and we'll get into what that means and why it's significant and the rate of learning. So starting off with the history of synaptic plasticity. So Plato, as you might have heard of that guy before. He's a pretty famous ancient Greek philosopher, and he described the brain as a wax block that may vary in size, cleanliness, and consistency in different individuals. And we'll see how that has some truth to it later on in the workshop. And then, however, before the 1950s, researchers thought the brain was static, so it didn't change. It was just very unchanging. And after adolescence, and so that's your early years. And then Donald Hebb suggested that neurons fire together, wire together. So this means that if two neurons fire at the same time, their connection will get stronger, and we'll add on to that very soon. So moving on to our very first little quiz. So this is part of the content that was covered on the very last slide. So the question is, what did Donald Hebb suggest neurons do? Let's see. It was a short little quote. Anyone got it? If not, that's totally okay. Yes, that is nice job, Catherine. Awesome. Yeah, so neurons that fire together, wire together, neurons fire, neurons wire. That's very accurate. Awesome. Good job. Okay. Next slide.
All right, so now we're going to move away from history and move into the actual neuroscience behind this. So we'll start with an intro video on neurons.
We can't hear the audio, but is there a way to get that?
In this installment, I will discuss the neuron. This is a brain. Estimates vary, but right now the best guess seems to be that our brains contain around 85 billion neurons. The neuron is a nerve cell, and it's the primary functional unit of the nervous system. This is a generic image of a neuron. Neurons actually come in all shapes and sizes, but this is the prototypical version of a neuron that you'll often see in a textbook. The structures extending from the left side of the neuron that look a little bit like tree branches are called dendrites. Dendrites are the area where neurons receive most of their information. There are receptors on dendrites that are designed to pick up signals from other neurons that come in the form of chemicals called neurotransmitters. Those signals picked up by dendrites cause electrical changes in a neuron that are interpreted in an area called the soma, or the cell body. The soma contains the nucleus, which contains the DNA, or genetic material of the cell. The soma takes all the information from the dendrites and puts it together in an area called the axon hillock. If the signal coming from the dendrites is strong enough, then a signal is sent to the next part of the neuron, which is called the axon. At this point, the signal is called an action potential. The action potential travels down the axon, which is covered with myelin, an insulatory material that helps to prevent the signal from degrading. The last step for the action potential is the axon terminals, also known as synaptic buttons. When the signal reaches the axon terminals, it can cause the release of neurotransmitter. These purple structures represent the dendrites of another neuron. When a neurotransmitter is released from axon terminals, it interacts with the receptors on the dendrites of the next neuron, and then the process repeats with the next neuron.
All right, so that was a bit of an intro to neurons and how they communicate. So now let's get into synapses, which is really the meat of how neurons communicate. So chemical communication is more common than electrical communication. So neurons can communicate through chemicals and electrically, but chemicals is the one we'll cover today since it's a lot more common. And so the steps for chemical neuronal communication are that first an action potential from the neuron's axon, which you saw in the video, reaches the synaptic terminals. And so as covered in the video, an action potential is an electrical signal that travels on a neuron's axon and achieves the neuron to send a signal to another neuron. And so the action potential causes ion channels to open and close, which causes neurotransmitters to be released from the terminal. And so that is a lot of information, but hopefully, so just to cover it again real quick. So the action potential is an electrical signal that travels down a neuron's axon. It reaches the terminal, and then that activates ion channels to open or close, which then leads to the release of neurotransmitters. And neurotransmitters are chemicals that bind to the receiving neuron's receptors after traveling through the synapse to make it more or less likely to fire. And so you've probably heard of neurotransmitters before, and some famous ones are dopamine, oxytocin, things like that. And maybe ones you haven't heard of, like GABA or glutamate. There are tons of neurotransmitters, and a lot of them haven't even been discovered. So they're very, very cool. And then once the neurotransmitters leave the terminal, they're in the synaptic cleft. And so that's where they're going to travel over to the other neuron's dendrites, and then to bind to those receptors. And then so once they actually bind to those receptors, then the message the neurons are communicating is made either stronger or weaker. And that is essentially synaptic plasticity, but we'll get into that a bit more in a second. So moving to our second pop-up quiz question. Don't be nervous. It's just to check in with you and see what you've been learning. So this question is, what chemicals are released into the synaptic cleft and bind to other neurons? An example would be dopamine. Yes, Melanie Davis. Yes. Awesome job. Yes, Vivian. Yes, we're learning. This is amazing. Yes. So neurotransmitters are going to be those chemicals. Great job, everyone.
All right, so now that we have a good handle on neurons and their synapses, we can get into how communication of those neurons can lead to changing how your brain is wired. So synaptic plasticity is a process by which the synapses between neurons change structurally and or functionally. It also controls how effectively two neurons communicate, meaning it can change the pathways of neuronal communication to be stronger or weaker. On the next slide, we'll cover why synaptic plasticity is important. So synaptic plasticity describes the ability for our brain to change throughout our lifetime, and that's the basis of learning, memory, and adaptation. You might have felt like a new person during the pandemic, and that probably results from you adapting to the new situation, which was the pandemic. Another example of synaptic plasticity at work is in taxi drivers. Studies have shown that their hippocampi, the structure in their brain that partially deals with spatial navigation, are larger in taxi drivers. And synaptic plasticity is involved with a wide range of things, including forming our earliest attachments to habit forming behavior throughout our lifetimes. For example, if chewing on your nails helps you release stress, your neurons that connect to your nail biting pathways will be increasingly activated when you're stressed.
Yeah, so now that we know a basic, like why synaptic plasticity is really important, now we're going to cover how it actually works. And so the main feature, and this will be really important in the next slide, is the gradual increase or decrease in postsynaptic receptors. So those are the structures that receive those neurotransmitters, the chemicals that go through the synaptic cleft and bind to the receptors on the receiving neuron. And that triggers an electrical signal, which we covered before, is the action potential, if you remember that. So as you can see in the picture on the bottom right, neurons adapt to sustained simulation. And so that means they can alter the amount or type of receptors they have. So certain neurotransmitters will be more likely to bind. And that's essentially synaptic plasticity, is the neurons adapting to different situations. That's your brain adapting. And so your brain is literally rewired in certain instances when you learn, take in new experiences like the pandemic, etc. So moving on to the next slide, we have our third check-in quiz. So I hope you all are learning a bit. And so this question is, what is the main feature of synaptic plasticity? Who can tell me? The main feature is the very first thing covered on the last slide. Has something to do with postsynaptic receptors. Let's see if anyone remembers. Yes, Vivian, awesome job. It's like verbatim. I don't know how you remembered that. Maybe it's in your notes. That's awesome. Yeah, so yeah, it's the gradual increase or decrease in postsynaptic receptors. So that's the neurons adapting and changing based off of new experiences. And so your brain is rewired, and that is synaptic plasticity. And obviously it's not on that scale with every new experience, is like an entire rewiring of your brain, but the neurons do change, and that is synaptic plasticity. So moving on to LTP and LTD.
So yes, LTP and LTD. These are two ways that your synapses can change. So LTP is long-term potentiation, which is a strengthening of synapses, and it's thought to be crucial in memory formation. And then LTD is long-term depression, and that's a decrease in postsynaptic strength. And it's also thought to be involved in memory as well as learning. So one way that long-term depression is involved in memory and learning is that it's thought to help in the removal of old and useless memories and skills, such as decreasing drug use urges in rehabilitation stages, or unlearning the wrong way to do something. This can be in like swimming, if you're swimming the wrong way. Long-term depression would help in removing the procedural memory, that wrong procedural memory from your mind. Long-term potentiation would help you retain the newly learned way to swim. So an example is that you can see in rodent studies is that you can see synaptic plasticity in the nucleus accumbens, and how that is central in long-lasting cravings and relapse in rodents addicted to meth. And this is because the nucleus accumbens is often labeled as the pleasure center of the brain, and it's located in the basal forebrain.
Yeah, so the rate of learning, so that's an interesting topic involved with synaptic plasticity. And so research has shown that STDP, which stands for spike timing dependent plasticity, which is kind of a mouthful, it's central in memory formation in neural networks. And so the strength of the synapse is either strengthened or weakened depending on the timing of the firing of pre and post synapses. So that's another layer of synaptic plasticity, along with the changing of the amount and the receptors on the receiving neuron and things like that. This is another layer to synaptic plasticity. And so the next slide will focus on a study that contributed to this research. So as covered in the last slide, the timing of firing matters a lot. And so I'll kind of explain what that means. So the study measured the time difference in the pre and post synapses firing. So it measured that if the timing of the firing of the pre and post synapses in two neurons was less than 0 milliseconds, they found this by subtracting the firing time of the post synapse by the pre-synapse to get that, above or below 0 milliseconds. And if the difference was less than 0 milliseconds, this tended to lead to LTD or long-term depression. And that, again, is the weakening of synaptic connections. And if the difference of the firing time of the post minus the pre-synapse firing was above 0 milliseconds, this was shown to lead to LTP or long-term potentiation, or the strengthening of synapses, which was also covered in the last slide. And so, or a couple slides ago. And then you can see all that information in the image. So if you look at it, you can see that if it was all those little dots that are to the left of that middle line there, that's below 0 milliseconds. And so that's going to be LTD or long-term depression and that weakening of the synapses. And then everything to the right of it is going to be above 0 milliseconds of the difference between the pre and post synaptic firing. And so that's going to lead to LTP, long-term potentiation, and the strengthening of the synapses. And I hope that makes sense. If you have any questions, feel free to put them in chat. But for now, we'll move on to a case study.
Yeah, this is a case study we wanted to talk about. So the case study was conducted by Dr. Psyche Louie at Wesleyan University. And so she's also, she's a neuroscientist, but she's also an accomplished violinist. And she found that the right hemisphere motor cortex, which controls the left hand and is used to precision by violinists, has more structural connectivity in those who play the instrument. And also, like, the younger the violinist starts playing, the greater the structural connectivity. And this increased structural connectivity is caused by synaptic plasticity and mechanisms like long-term potentiation, which allow for better communication between neurons.
All right, so it is time for a Kahoot. So let's get started. This has some questions about what we've covered so far in the presentation. And so Kahoot is pretty famous, so I hope the majority of you have played it before, but if not, it's essentially a series of questions about what we've covered so far in the presentation, and you all answer, and yeah, it's a bit of a healthy, competitive game. So you'll put in the code, and then we will get started. So just try to put, yes, I love Kahoot. Awesome. I'm very glad about that. Yeah, it is really, really fun. So the code is 8837566. And so to do that, as it says on the screen, just go to kahoot.it and it should lead you right in. We've already got people flowing in. Awesome. Yeah, this game is really fun. So hopefully you all enjoy it. But yeah, as everyone else is doing, just put your first name. You can put your last if you'd like. Your initials is fine if you don't want to expose yourself or anything. All right. And then once enough people join, we'll go ahead and get started. Rely on your memory. If you took notes, you can look back at your notes, but it should be really fun. And don't be stressed out. I know I can get a little stressed out when I'm playing Kahoot just because of the competition aspect of it, and you have to be fast with your answering and everything like that. I know it can get stressful, so just keep in mind that it's a check-in for your learning, and it's not cutthroat or anything like that. Just a bit of a check-in to see what you learned throughout the presentation. All right. Oh no, someone dropped out. We had 12 and then we have 11. Come back. Whoever wants to play, you don't have to if you don't want to. It's all good. But we'd love to have as many people as possible. Maybe another minute or so for those who'd like to join. All right, it doesn't look like anyone else is going to join. So I think we can go ahead and get started. So it's all about what we've covered so far. All right, so what did Plato describe the brain as? Yeah, pumping up the music. Yes, a wax block. Yes, 100%. Awesome, everyone. All right. Next question. So who suggested that neurons that fire together wire together? This was one of our check-in questions. Maybe Obama, York, who knows, who knows? Yes, 100% again. Look at you all go. That's awesome. Holy moly. Some healthy competition. All right, next question. So which part of the neuron receives signals from other neurons? So this was mostly covered in the video, but it was also on some of the slides. Yes, dendrites. Awesome. Great job, everyone. VB popping off with the answer streak. Very nice. All right, what is the soma? Yes, good job. Oh, looks like, yes, I believe we will be able to share the slides, so we'll be able to do that for you all. Someone was asking in chat. All right, next question. What does an action potential cause in a presynaptic neuron? I don't know. It's a toughie. Yes, nice job, everyone. All right, so this question is, once released from a presynaptic neuron, what is the fate of a neurotransmitter? Nice, that one was quick. You all learned a lot. This is awesome. All right. Let's see, we're about halfway through. Push through. What is synaptic plasticity? Nice, 100% again. Look at you all go. This is awesome. So what structures in postsynaptic neurons underlie synaptic plasticity? Could be the soma, hippocampus, postsynaptic receptors. Awesome. So the soma is going to be the cell body. So it's going to be the postsynaptic receptors that are going to be involved in that. All right. So what does more postsynaptic receptors and a postsynaptic terminal facilitate? Maybe a water bottle? Who knows? Stronger response. Someone put water bottle. I don't. It's a good guess, but not quite the answer. All right. Very healthy competition. Love to see it. So what is LTP? Yes, nice job, everyone. Let's see what the next question's all about. So what is the nucleus accumbens responsible for in the brain? Nice, that one was really quick. Y'all have fast fingers, awesome. Nice one's true, false. Just to read it out loud, the STDP is not central in memory formation in neural networks. True or false? That is false. Good job, everyone. All right, so what part of the brain has more structural connectivity in violinists? This was the case study that we did at the latter part of the presentation. Very nice, everyone. The right hemisphere motor cortex. Awesome. They're going to be moving around. Well, their fingers will be. All right, this is kind of just a fun question. What do you all think? What is the best? What comes out on top? Obviously, there's many more genres, but for the sake of this Kahoot, all of them. A lot of you like pop, though. Yeah, every music genre is amazing, and it depends on your individual pathways, your neural pathways to see which music you like. It could be any of those. All right, let's see. Nice, nice. 14 out of 14. Awesome. Another 14 out of 14. Very nice. All right, who came out on top with another 14 out of 14? Melanie, awesome. Claps for Melanie. Very awesome. And everyone did incredibly. That was not the easiest Kahoot in the world, and you all absolutely popped off. So congrats, everyone. And that wraps up the Kahoot. So back to our presentation. It looks like the screen is frozen. I don't know if we're just chilling on one screen but it looks like it's frozen. Is it frozen for you, Pavan?
Yeah, it is.
Okay. Yeah, I didn't know if that was just my Zoom being weird. Yeah, so hopefully we can get back to the presentation. We just relish in the absolute W's of our top three.
Yes, just a second, guys.
But yeah, I guess in the meantime, if you all have any questions, feel free to put those in chat. All right, so now we're back at the presentation. So yeah, here are just some resources to learn more. There's a ton more than this as well to learn about the brain, synaptic plasticity. There are research papers galore. But here are some of edX. That's a really cool online course platform. Khan Academy, I'm sure you all have heard of. Coursera is another online course platform. The Neuroscientifically Challenged 2-minute neuroscience series is from the video that we watched earlier is from that resource. And then Brain Facts textbook, MIT Intro Neuroscience course. All of those are very, very cool resources that you can check out. If you want to read research papers, there's a lot of databases and just there's like an infinite amount of research papers, and synaptic plasticity is a really cool and popular topic. So there's a lot you can read up on that. Some resources for research papers include PubMed is a very large open source research database for papers. Scopus is another one. There's a ton out there for you. So moving on to wrap up our presentation. So we hope you enjoyed. This was a lot of fun for me. I hope it was fun for all of you too. Our emails are right there. So Erin and Pavan at simplyneuroscience.org. If you have any questions, you can also throw them in chat if you would like. Can all the resource links be posted in the Google Classroom? Gerline, is that something we can do?
Yeah, we'll post the slideshow onto the Google Classroom and everything will be obviously in the slideshow.
Awesome. And yeah, we'll try to get that shared for you all. But yeah, that about wraps it up. Thank you all so much for coming, and then hope you have an amazing day. And if you have any questions, feel free to put them in chat. You're very welcome. That was a lot of fun. I hope you all learned a lot. And maybe you can even notice some examples of plasticity in your own life. I bet adapting to the pandemic applies to all of you. Yeah, of course. Here are just some sources that we used for images and things like that in our info in the presentation. Hey, I'm glad you enjoyed it. You're very welcome.
Okay, and so thank you so much to our BRC team for that awesome workshop. I'm sure you guys learned a lot. It seems like you definitely did because that Kahoot was so competitive. That's everything for today's kind of session of the Cerebral Cortex camp. Thank you to everyone who joined us for today and worked so hard this morning and paid such close attention to our workshop this afternoon. Tomorrow morning, we're going to be starting with another workshop followed by lunch and then a work period for your outcome projects. If you have any questions, feel free to stay back. Otherwise, we will see you guys tomorrow. Bye, everyone.